Remember 2020? Masks were in short supply. Group gatherings were banned. And we were all stuck at home in lockdown. Vaccines from Pfizer and Moderna put an end to all of this. But how? For decades, vaccines have worked the same way, by introducing a dead or weakened virus into the body. When viral particles enter our body, our immune system springs into action to protect us, even if these particles aren't harmful. Different types of lymphocytes, which are white blood cells, respond to this threat in different ways. B lymphocytes respond by producing antibodies that bind to the chemical markers on the surface of the pathogen, called antigens. This marks the pathogens for destruction by other white blood cells. T lymphocytes kill virus-infected cells by recognizing viral antigens displayed on their surfaces. Some of these B and T lymphocytes become long-living memory cells, which produce a faster secondary immune response when the same virus reinfects the body. So, in other words, we are tricking our immune system by sending in a dummy of a virus in order to train it to respond better and faster when the real virus infects us later. Then came a new approach: mRNA vaccines from Pfizer and Moderna. These relatively new vaccines contain molecules called messenger RNA, or mRNA. Messenger RNAs are a type of RNA which are nucleic acids. Like DNA, its structure is composed of a phosphate backbone and 4 nitrogenous bases: adenine, uracil, cytosine, and guanine. Instead of thymine, which can be seen in DNA molecules, RNA has the base uracil. These bases act as a blueprint for building useful macromolecules. Another difference is the sugar molecules these nucleic acids contain. While RNA contains the sugar ribose, DNA contains the sugar deoxyribose, which lacks an oxygen atom compared to ribose. Lastly, mRNA has a special role. It carries genetic information from the nucleus to the ribosomes, where the information is used to piece together amino acids to build proteins. Thus, protein synthesis occurs. In these vaccines, the mRNA is engineered in a sequence of bases, which code for the large antigen found on the virus's surface called the coronavirus spike protein. Once the mRNA enters a cell, ribosomes read the sequence in groups of 3 bases called codons. Each codon matches a specific amino acid, delivered by a transfer RNA. Piece by piece, the ribosome builds a chain of amino acids into the exact sequence of the coronavirus spike protein. This harmless protein is then exposed to the immune system, which responds and remembers, just as if the real virus had arrived. Compared to the old method, this new method has huge advantages. Firstly, only a harmless part of the protein enters our body, greatly reducing the risk of side effects. Secondly, this new method is easily adaptable. Even if new variations of the virus emerge, a mild adjustment to the mRNA will suffice to make the vaccine effective again. Lastly, since there are no live viruses to handle, the production process is much faster, safer, and cost-effective. During this horrific pandemic, scientists and researchers all around the world worked night and day to save countless lives. Their success with mRNA vaccines didn't just bring our lives back to normal. It opened the door to a future where science can protect us faster than ever before.